carbohydrates, the naming of the ketones and the aldehydes, and what kind of structures are present in those particular diverse molecules, particularly as we add carbon chains to the elongating backbone of the carbohydrate. We have these chiral centers, which then give us a number of different molecules, the imprimers and antimers, in regards to the structural alliance and the rotation or movement of the hydroxyl position, either to that left or that right hand side, as well as that D or L configuration in regards to that carbon oxygen hydroxyl, the carbon hydroxyl position being an indicator of whether it's D or L. We talked about stereospecificity and again the diastereomic component in L and D, and the number of chiral centers that are present in these carbohydrates given a simple formula. The D and L components in regards to that most distant second carbon from the functional group, whether it be the aldehyde or the ketone. And we can see, importantly, then how things can change and form imprimers in regards to having just simple changes in that hydroxyl positioning in relation to a reference compound such as glucose, where we see galactose and D-mannose being imprimers. We then looked at a little bit of the diversity of those particular carbohydrates in the aldehydes and the ketones. And then we looked at hemiacetals and hemiketals in regards to how we cyclize five or six membered ring. I should be careful about that. It's not 6 carbons, it's 5 or 6 membered ring because there's an oxygen involved there, as you can see in the formation of the cyclic form of D-glucose. Then we also looked at this anomeric carbon, this carbon number 1, and its issue in regards to having a rotational capacity, we call mutarotation. And then we had a look at a little difference between the alpha and the beta sugars in regards to the positioning of the hydroxyl form in the anomeric carbon number 1 position. We had a little bit of an insight into the structural configuration of these cyclic sugars in regards to the individual units becoming either chairs or boat forms and basically minimizing their steric hindrance. We also had a look at the chemical diversity of the hexoses, in this case the glucose backbone or glucose cyclic molecule, in regards to what we can actually diversify structurally with these. And they all seem to be following a very similar event component by first going through a glycosamine, then having that amine derivatized with an acetylation, and then that acetylation also then having capacity to have attract additional side chain modifications as we move through the structure, as we look at N-acetylmuric acid down there in the corner. We also have this capacity and this common chemical reaction within biochemistry from an aldehyde, carbonyl group, to a carboxylic acid or carboxyl group. So this is again a reaction we're going to see time and time again, an aldehyde to a carboxylic acid. And we can see that with these reductions of acidic sugars. We then talked about reducing sugars in regards to that anomeric carbon playing a very important role in its availability in this, but knowing that importantly that the reducing sugars can only occur in their linear forms, not in their cyclic forms. We then talked about the glycosidic bond, which is very important. We're going to have a look a little later on in glycosidic bond formation with amino acids, because amino acids become a central part of our biochemistry, but we can also now decorate these with chemical functionalities, including carbohydrates. We'll have a look at that a little later on. We're talking about different bond configurations, so you're going to have to understand different bond configurations and being able to draw those. And the examples that I've provided are the ones that you should know in regards to the beta 1, 4. Then we have the alpha 1, 2 beta. This one is a illustration of both a pyran and a furan being conjugated together by a glycosidic bond, a 6-membered ring and a 5-membered ring, but still having a 6-carbon backbone in this case. And then, as I said, the more simplistic version in regards to actually flipping over one of the particular pyrans, in this case a glucopyranose side, the final carbohydrate here being flipped over and giving us a 1-to-1 structural configuration. I introduced then also, and this is where we left off in regards to the 3-letter codes or numeral letter codes in regards to some of the basic carbohydrates. Now, I don't expect you to know these, but there'll be a few that you might become more familiar with, just the way we're basically working up the materials to understand processes of metabolism. This is where we stopped on Tuesday, so let's continue. So when we start looking at the most abundant carbohydrates in our planet, we're going to see these as very long polysaccharides, or they call them glycans. And they can be both homo or hetero polysaccharides, homo being repeated same units of the same nomenclature, where the hetero polysaccharides can be varying and branching. We'll have a look at those very shortly. But the carbohydrate that is most abundant in nature is cellulose, wood. Think about the wooden furniture, the trees, the plants. Cellulose is there. The interesting thing with cellulose, it has a beta configuration in regards to a beta bond, a beta glycosidic bond, at a 1-4 linkage. And this basically provides, some level of protection in cellulose itself. Because wouldn't it be amazing if we could just chew on a piece of wood and get all the sugars we need, because we can break down those particular carbohydrate backbones. But we can't. We don't have the enzymes to do that. What we do, though, is actually have enzymes in our bodies to break down the alpha 1-4 linkage, which we see in both starch and glycogen. Starch is a plant material also, a high energy source for us as well as for plants. But also glycogen, which is a metabolite product that we actually make to store sugars to reduce the osmolarity, and then have hopefully access to these individuals by having enzymes that break down those 1 and 4 linkages to give us basically the individual carbohydrate coming off, which we can utilize. So we see in common homopolysaccharide are commonly used for fuel storage and structural elements, as I said, we illustrate here with the homopolysaccharides of starch, glycogen, and cellulose, where the heteropolysaccharides, which we'll have a little bit more of illustration today, can be used as a kind of a cellular structure, support system. They're a little bit more decorated in regards to having attractions to other molecules such as proteins. And they're commonly called, those ones that are attached to actually peptides, are called peptidoglycans. So we're going to have a look at those in a particular time in the next couple of slides. But the interesting thing is with these polysaccharides, particularly what we see in cellulose, starch, and glycogen, people ask, how big is that molecule of glycogen? And it's actually a very poorly defined molecular weight. Usually, I think the nomenclature is that they have to have more than 50 subunits to become a polysaccharide. Go back to our previous lectures in regards to the hierarchy of molecules. And we actually use these to our advantage in regards to producing very complex homopolysaccharides, which then have this capacity to be able to be storage elements in starch. That's for plants, glycogen's for us. And the interesting thing is they're very heavily hydrated. So again, a lot of hydrogen bonds occurring in regards to basically stabilizing the individual structures. So when we have a look at glycosidic linkages in this case, amylose, we have basically a homopolysaccharide of glucose, and it has a alpha 1,4 linkage as it is illustrated here. And interestingly, we have a non-reducing end, so an end which is going to be in the number 4 position, then basically then a 1, 4 again, 1, 4 again, etc. So we see along here. But there will be an end point in regards to somewhere in the structure where there will be a carbon, an anomeric carbon, available for reduction, and this will be the reducing end. So it's a bit like what we see with amino acids in the N and C terminal. So we just call them non-reducing end and reducing end. So with these alpha 1,4 interlinkages, that's all okay in regards to producing a long strand of carbohydrates such as glucose. But the problem is there has to be a little bit more regimented structure to allow us to really get the best opportunities in regards to insolubilization or solubilization, depending which way we want to go, but also access to an enzyme system that's going to break down these particular glycosidic bonds and cause the release of the glucose moiety. This is where we might see branching occur. So here we have a long chain of 1,4s, but we might also see a branch point along these 1,4 constituents. So here we have the 1,4 constituents. But the carbon number 6 can also branch to another parallel polysaccharide chain, usually of a homopolysaccharide chain, in a situation of now connecting that carbon number 6 to carbon number 1, as we illustrate here. So here we have a molecule now that has long strains of repeating glucose units, mostly in the 1 to 4 unit, but now and then there will become a cross-linking component in a 1 to 6 bond. And again, the enzymes that are required to break these particular bonds are different. So an alpha 1,6 enzyme is different to an alpha 1,4. Now, these branching points give us some very interesting capacities in regards to really packing in the sugars in these long polysaccharide chains together. And this branching point and the combination of the homopolysaccharides produces this amylopectin. Now, amylopectin basically is just a long chain of alpha 1,4s, but every 24 to 30 residues, there is a 1,6 link. And what we actually get in the end is this type of structure occurring and basically allowing us to organize the carbohydrate strands. So here we have the 1,6 connecting those individual ones, and then we have the 1,4s with the long elongation like we see with the amylose. So again, here's amylose, just 1,4. And then 1,6 is coming in there particular to produce the amylopectin. What it really gives us is the capacity to kind of tie up the reducing ends and allow us spontaneous and multiple interactions with the non-reducing ends. So it gives a level of coordination, which we'll see in some other enzyme reactions, in regards to presenting a substrate that has multiple positions where it can be degraded or built, depending on which our energy requirements or processes are needed, in regards to elongation or shortage of these particular chains. So we might have an amylose coming in here and basically wiggling along a template chain, which is already established in the amylopectin. But it's these reducing ends and the number of these reducing ends that gives us the capacity then to we need to have one single enzyme, remember our kinetics, if you only have one substrate and an enzyme that does a lot more work than that capacity in the turnover rate, it's not going to be used very effectively. So we might have one enzyme and multiple substrate binding sites, which will then hopefully allow the enzyme to work a sufficient way to actually cause the degradation and the release of those carbohydrates as in the form of glucose. And that's free glucose, which we can then utilize. And we'll talk about that utilization of free glucose a little later on as we look at the metabolism of glycolysis and gluconeogenesis. So it gives us this capacity to increase the interactive coordination between substrate and enzyme, and this is established through the process of having structure in the form of the alpha 1 4s to give us the linearity, and the branching of the alpha 1 6s to give us that change. So glycogen, which, by now, if you've not had breakfast and you haven't eaten since about maybe midnight or something like that, most of your glycogen is now basically being depleted. When we are surplus in our carbohydrate intake, the first thing we do is repack the carbohydrates to produce glycogen. And that becomes then a stable source for us to basically chew out a little later on when the glucose levels start to deplete. The idea is that the body is trying to maintain a glucose concentration. So glycogen is extensively branched more than the amylopectin, where we'll see branching occur about every 8 to 12 carbohydrate residuals, in this case glucose. And the liver is where it's stored, about 7%. But we also do have some in skeletal muscles. And this is basically to give us a reserve of energy in the skeletal muscles if there's a flight or fight mechanism in regards to not having to contact the liver, move the carbohydrates in the bloodstream to the muscles to make them contract or give them the energy to contract by their degradation to make ATP. But we then have some in the muscles as reserve to allow us to have a spontaneous, hopefully very quick reaction. So these are branched molecules are in the same kind of process of the alpha 1 4 and the alpha 1 6 linkage, as we saw previously on the previous slide. And there are very specific enzymes for both their synthesis and their degradation, and we need to regulate those given, again, the balance of energy that we need in our bodies to allow us to actually form and function. So if we're going to be in a happy state, in the cellular happy state, where there's all the energy requirements being met, and that we have a surplus amount of energy in regards to forming the broken down or obtained carbohydrates, that we're going to then synthesize these kind of molecules such as glycogen, because we're going to keep those a little later on and store those. But we can only store so much glycogen. There's other forms of metabolism which we switched on if the energy abundance is much higher. We'll have a look at those a little later on, and then we can actually cipher those into either make amino acids or fats, and we'll talk about those. But the important thing is also this degradation, because we want to maintain a carbohydrate level within the circulatory system. So therefore, we need to make sure that we have very active enzymes to allow us to actually cause that degradation and effectively in regards to getting that sugar or the single sugar moieties back out into circulation. Though they may be present in the liver and the skeletal muscles, there's no storage of carbohydrates in the brain. So you've got to make sure you have a constant flow of carbohydrates or other moieties. And we're going to come to see those other moieties called ketone bodies in regards to metabolism as a alternative to glucose. And again, we've got to be very careful about utilizing those types of compounds, because there is some diasteric dramatic effects if we continually not feed our brain with glucose and basically feed it with ketone bodies. So glucose becomes an essential part for our existence in regards to feeding our brain. And our brain through evolution has grown because our body has become so efficient in the issue of capturing glucose or making glucose and storing glucose and degrading glucose. So again, that's one of our part of our evolutionary advancement. If you look at a jellyfish, though it has similar mechanisms metabolically, the problem is that the jellyfish does not have a very good storage system for glucose. So again, the development of its own neural system and its central nervous system is somewhat limited in regards to diffusion as well as the production, storage, and utilization of glucose. So glycogen has become a very advanced, molecule in evolutionary terms because of this numerous non-branching ends and only one reducing end, which basically allows us then to concentrate on the numerous new non-reducing ends to give us then degradation. So we can actually release a lot of carbohydrates in a very, very, very short time from a large molecule at multiple surfaces. We also see dextrans, which are a very large carbohydrate. If you're doing size exclusion chromatography, we use dextrans as a calibration because they're so large and complex. They are usually retained on the column. They're usually first not to be retained on the column. They come out very quickly in the void volume or near the void volume, and that's how we get a calibration of the column to know what a column volume is in the sense of fluid movement. These come from bacteria and yeasts, and they're a polysaccharide of a 1,6 system, but this time they have varying branching systems. So again, at the 1,3, 1,2, and 1,4, so very diverse. So a lot more like a net, fish net, in regards to the types of branching and interlinkage between the strands of the polysaccharides. Now, when we look at the structural role of some of these polysaccharides, particularly on the homose polysaccharides, we see cellulose as one of the major contributors which we become familiar with. And as we know, cellulose, it was basically, it floats. There's an air volume weight ratio difference. It's also insoluble in its linear unbranched form in water. So again, it has a capacity to deflect water. And the interesting thing is that all the bond structures are in the beta configuration, so beta 1,4 bonding in regards to being that kind of strictly upturned one. So this provides us some interesting properties and physical properties regarding wood. It gives it the issue of the hardness and different types of wood in regards to the strengths basically reflect the types of bonds and the majority and minority of bonds in regards to the strength and capacity to be flexible. So again, a bit like what we saw with hair in regards to the disulfide content, this time it's reflecting on the bonds themselves, the glycosidic bonds, and the makeup of the glycosidic bonds to give you different characteristics of the wood. We already know we can't chew wood and get the carbohydrates out, but there are insects that have the capacity to consume wood to make a pulp and then use enzymes that they don't actually produce. They have microorganisms in their microbiome in their gut, which basically produce these enzymes as a secondary product. So there's a symbiotic relationship where these microbes are given an environment where there is food available, certain nutrients available, and they secrete enzymes which the insect uses, such as termites, to allow you to then basically access the carbohydrates locked away in the beta 1,4 configuration of the wood material. So unlike us, we can't consume wood or grass. Sometimes we will actually consume plant material that is obviously got a lot of cellulose in there, and that becomes what we call fiber or roughage. We can't digest that, which then allows maximal passage through the GI tract. People have thought about, why not put symbiotic microorganisms in human beings as a part of a probiotic. That might be possible, but No one's done it so far, but I'm sure people are thinking about it in regards to being able to access this most abundant carbohydrate on the planet. Is you start thinking in regards to what we are going to be as humans in 150 years' time. Now, the next most abundant polysaccharide is in insects, and that's known as chitin. And chitin is actually the hard material that makes up the exoskeleton of the insect. So it's a N-acetylglycosamine polymer in beta linkage. So a bit like what we saw previously with our starch, our cellulose molecule in regards to 1,4 configuration. But noticing this time we have an orientational difference in regards to our repeating subunits. So there's an orientational difference in regards to being up, down, up, down, up, down, and giving you this unique, or not so, well, it's a unique bond configuration, together with in position number 2, that acetylation on the amine component of the six-membered ring. So there's the amine, there's the acetylation. The combination of the bond configuration as well as the acyl acetylation or the N-acetylation and the beta bond gives us the capacity to have strength. You might have heard of a compound called shellac. And shellac is a solvent that is actually painted on wood to give it a very, very, very hard surface, like a varnish. They make synthetic shellacs these days, but they still use, I think there's some shellacs that still use beetle exoskeletons or beetle shells and dissolve them in a solvent such as turpentine. And you can paint those on, and it gives you the same characteristics in regards to strength. Now, as I said, we've seen these now folding polysaccharides or these particular characteristics, which are in principle very similar to what we've seen in proteins and peptides. So keep this in mind in regards to the parallelism that we've seen. And again, they're also stabilized by the same forces of physics, the weak van der Waals forces, the electrostatic interactions, particularly between the various chains we saw with the amylopectin, as well as also we saw with cellulose, and then also with the previous structure in regards to the amylose structure and, as I said, also the amylopectin, dextrans too. But hydrogen bonding in this case is actually one of the most important and largest influences because of the simple characteristic of the molecule having so many hydrogens and so many hydroxyl functions. So again, we have then this role of water playing a particular role in stabilizing these particular structures. A little like what we saw previously with the Thi-Si bonds in regards to peptide backbones, we also have capacity to have these measurements of Thi-Si angles in the glycosidic bond. And with these particular components, we can start realizing that there's going to be defined structures that we could establish. But unlike what we've seen with the peptides and proteins, these structures unfortunately can be very, very, very weak in regards to their stabilization. So dextran, which has a lot of these repeating 1,6,1,3 bonds, and we're going to have some kind of angular rotation. And ultimately what we're going to see is maybe some kind of like a beta sheet formation occurring. But when we get to like the 1,4s, whether it be in cellulose or in amylose, we're going to see the potential to form a alpha helical kind of structure, a repeated component in regards to a circular component structure. With, again, like we saw previously with the alpha helix, the residuals basically playing an interface role in stabilization of their neighbors, in the number of turns, and then also issues of termination and starting a particular structure. But again, these structures are only stabilized in most by hydrogen bonding at a higher number and give it certain strengthful components. But we've got to be very careful. If we start changing the chemical environment in solvation or in heating, we can start breaking those particular bonds. And that's why, as we see, when we see wood burn in regards to breaking these particular thermal labile bonds, which then generate energy, and basically then we're left with a carbon constituent such as charcoal, which is coming from the carbon backbone of the, in this case, of cellulose. So again, we've got to kind of think a little bit more in regards to what we understand about woods and oxidation and heat generation, given that we can access and break these particular bonds in this particular structure configuration, and it's going to change as we oxidize or incinerate the material. Now, the peptidoglycans a little more complex, and a lot of interest, obviously, in these guys, given their situation that we're seeing now with interactions of foreign elements such as viruses on a cell wall. So peptidoglycans are these kind of heteropolysaccharides that can be present in a bacterial cell wall, and they provide the capacity to provide a distinct and different extracellular matrix, which allow things to bind to them. And how they actually do this is they have these kind of recognitional sites in regards to the addition of amino acids and these carbohydrates. And as we know, there are going to be some enzymes, such as lysozyme, which is secreted in tears and various secretory material, has the capacity actually to degrade these types of carbohydrate interlinkage and weaken the cell wall of the bacteria. So this becomes very, very important in regards to a primary immune response, using enzymes to hopefully erode or decay or weaken bacterial cell walls to allow you then to actually attack those. But there are also other particular components, as you've seen with the N-acetylglucosamine and the N-acetylmuric acid, in regards to also being susceptible. But these guys also play a role, which we'll see a little later on when we look at viruses. So there's going to be a peptide, a common structure in regards to what we see here with this alanine, glutamine, lysine, alanine configuration, then interactions, see how they have a bond formation with the alanine and the N-acetylmuric acid. But then there's a lateral component of a repeating glycine moiety to give you that kind of backbone structure to allow you to basically, again, produce these very decorated carbohydrate structures. So they're cross-linked with short peptides. They have alternating beta-1 and 4 interlinkages with the glycosamine, the N-acetylmuric acid residues. And importantly, as I said before, we have this known sites in regards to their degradation using enzymes that we can work with. And this becomes then a potential for us to wave off and destroy any foreign bacteria that gets into our bodies or near our surfaces, particularly like secretory components on the skin. Agar, you might have used agar in the laboratories. I'm pretty sure you might have made some agar plates. Amazing stuff agar is in regards to it has this capacity to hold enormous amounts of water and change its physical attributes in relation to its hydration. And again, if you put too much too much water in, not enough agar, your gel doesn't set, or it's kind of still fluidy but still viscous, or you put too much in and it changes the characteristics in regards to being a lot more fragile and breaking the agar gel. Now, agar itself is actually coming from an extraction from seaweed in most cases. So we see these as a part of the seaweed cell wall. And the interesting aspect in this structure is that it contains repeated moieties of D-galactose, which are sulfonated. And again, this sulfonation becomes very important in regards to enabling you to produce a three-dimensional structure. So there's two main polymers in seaweed, as we see. So we have agrose here, which is this very simple 1,4 with the sulfonation. And then we have the agropectin, so very similar to what we saw with the amylopectin, and a branching system combination, this time with a beta 1,4, to give you that type of extended structure with multiple reducing ends. It is this agar pectin component with the multiple reducing ends, as well as the layer upon layer structure, which gives the capacity for the agar to actually trap molecular water. Because what it actually does in its structure, as we saw similar to the amylopectin, produces a double strand which basically winds around itself, and these parallel upon parallel strands have that capacity, and it's going to basically repeat every 3 carbohydrate residuals. That repeat, that cyclic component, provides a small cavity to trap molecular water. And again, this is where we see the properties of basically agar playing a role in its structural entirety to allow you to capture molecular water, and depending on how much molecular water, changing the property, extension, or contraction of the alpha helix itself. So again, changing the strength or tension of the agar solution that you might be making up. So that's a good illustration, and you can extract agar quite easily from grinding up seaweed material, kelp also, in regards to getting that type of material and looking at. But you can buy it in most places in regards to like a bacterial microbiology laboratory. We use it continuously. So we've got a number of polysaccharides that we have looked at, from starch, glycogen, glucose, chitin, dextran, peptidoglycan, agrose. We haven't gone so much on the gluconates in regards to these particular molecules. We'll have a little bit more look at these in a little second. But the idea is, look at this, homo unit subtype, and then starting into the more complex constituents which have peptides attached to them, and small proteins and sulfonation occurring. So again, you're expected to understand some of the differences and some of the characteristical differences of these particular carbohydrates, and maybe some of their significant roles in regards to what they actually do. The size, as I said, is not very well defined in many of these particular constituents. Agrose is usually very small, where you can have amylopectin to be extremely large in regards to its molecular mass. So you need to just be aware of these and be able to identify some of the characteristics and roles that these carbohydrates play. So I'll give you some terminology: the monosaccharides, the oligosaccharides, the polysaccharides. We looked at ketones and aldehydes. We looked at ketoses and aldoses, emprimers, enantiomers, the hemiacetals, the hemiketals, the furans and the purans, mutarotation, reducing sugars, polysaccharides, starches, cellulose and glycogen, the peptidoglycans, and agar. So, some of the things you're going to be expected to know and do is, naming some of the structures of sugars, and we've gone through some of those, and you'll become more familiar with those. You'll hear them time and time again. So, I want you to start being able to draw, structures of glucose in its linear and its cyclic form, such also as fructose, because they're going to become common elements when we start looking at glycolysis and gluconeogenesis in about a week and a half time. I think it is a week a bit. The different bonding components in regards to the various 1,6, 1,4, 1,3. But again, once you start knowing the nomenclature system of the carbohydrates, you're going to be familiar when I say, what's an alpha 1,4 bond, you're going to be able to identify the individual 1 carbon, the anomeric carbon, number 4 carbon, and see the orientation in regards to whether it's an alpha or beta, and draw it appropriately. And then we just got some examples, as I said before, in regards to understanding the structure of cellular starch and glycogen. I'd leave it at those three, and we'll take it from there. Okay, so we've kind of finished that particular lecture, and we're now going to go on to having a look in regards to the form and function of these particular proteins in regards to their utilization in the body. We've talked about proteins as enzymes and proteins as structural elements. We've talked about proteins as a functional moiety, as a carrier, such as hemoglobin and myoglobin. But we are now going to have a look at what happens with some of these carbohydrates. What's their role, and why do we need to have these in present?